the next set of topics that we're going to be delving into in the course are all related to reproduction and genetics and passing on of traits and characteristics. And we're going to begin this discussion at the cellular level by looking at different ways that different types of cells can divide and produce new cells. Since the evolution of cellular life some 4 billion years or so ago, the continuity of life, the production of new cells to replace old cells, depends on cell division. And depending on what type of organism you're talking about, that cell division is going to occur in different ways. Simple cells can divide and produce new cells by a relatively simple cellular mechanism, while cells that have more complex structure are going to have to rely on a more complex mode of reproduction. That makes sense. So what we're going to do in this discussion is look at the three main types of cell division, and we're going to focus most of our time on the types of cell division that are occurring in eukaryotic organisms rather than prokaryotic organisms. So the three types of cell division mechanisms that we're going to be talking about are binary fission, mitosis, and meiosis. Binary fission is a simple type of cell division that's used in prokaryotic organisms to produce new daughter cells. And it's a simple form of cell division, and it can be used because these organisms are structurally and genetically simple. Now, what do I mean by that? Well, we've talked before in the course about the fact that prokaryotic organisms lack internal compartmentalization, so they don't have internal organelles. They have one common cytoplasm, and all of their biomolecules are present within that same cytoplasm. In addition, we've talked about the fact that their DNA is found almost always in the form of one circular DNA molecule and maybe a few extra chromosomal plasmids that are not essential for life. So genetically speaking, from the perspective of their DNA, they're very simple. Binary fission is the means by which prokaryotic organisms produce new organisms of the same species. So this is propagation of the species that we're talking about, and the daughter cells are going to be identical clones of the parent cell. So this is a form of asexual reproduction. Now, when will bacteria or archaeal cells undergo binary fission? Well, primarily they're rate limited by nutrient availability in the environment. So as long as nutrients are readily available and wastes have not accumulated to toxic levels, these cells are going to be dividing. So for example, if you have E. coli growing in culture in the laboratory and you provide them with plenty of nutrient broth, they will undergo binary fission and divide every 20 minutes. And so essentially the population doubles every 20 minutes. Right, so it's a very simple mode of cell division. So let's just look very quickly at what that would look like. So here's a prokaryotic cell, let's say an E. coli, that has already replicated its DNA. So just to make clear what we're looking at here, which may be not quite so obvious, we started with a cell that had one DNA molecule, and the way that these organisms sort of keep track of and control the separation of their DNA once they've copied it is they use this very interesting little trick. They take their DNA and they attach it to the inside of the cellular membrane using proteins. And so the chromosome is actually connected to the inside of the cellular membrane. So before binary fission would happen, what would happen is that this DNA molecule would be copied. And so essentially we'd have two circular DNA molecules that are sort of intertwined and attached right next to one another at this inner surface of the cell membrane. And so what then happens is that this one common cell that now has two copied DNA molecules starts to grow and elongate. And the way that it does that is by inserting new cell membrane and cell wall here in the region between the two attachment points of the DNA. And so by inserting new cell membrane and cell wall here, that essentially pushes the copied DNA molecules away from one another. And so as the cell continues to grow and elongate, eventually those DNA molecules end up on opposite sides of the cell, and division of the cytoplasm and synthesis of new cell wall occurs here at the midline, and we end up with two daughter cells, again, both of which are identical copies of the parent cell that we originally started with. Now, binary fission is used by prokaryotic organisms, so bacteria and archaea. We should note that it is also used by some protists. So there are some protozoan organisms that also divide by binary fission. We're not going to look at them. They do weird things with their genomes, and so they're sort of an exception to the rule. But in general, binary fission is limited to those very structurally simple organisms in the prokaryotic domains. Now, once we move on to more complex cells that have that extensive compartmentalization and organelle structure, as well as a more complicated looking genome, binary fission is no longer sufficient to serve those cells' needs to accurately and with high integrity segregate the cellular contents into two daughter cells. And so a more complicated mode of cell division evolved, and that's mitosis. So mitosis is a complex cell division process that's used by eukaryotic organisms to ensure that their multiple duplicated linear chromosomes and all of their cytoplasmic organelles get appropriately segregated during the process of cell division. So eukaryotic organisms, what does that mean? Well, we know that that encompasses yeast, protists, plants, and animals. And so you can see mitosis occurring in single-celled eukaryotes like yeast. And again, like we saw in prokaryotic organisms with binary fission, the purpose of that mitotic event is going to be propagation of the species. When a yeast cell divides by mitosis, it produces two new daughter cells and so essentially continues on that population. Now, like we saw with binary fission, mitosis is going to be a means of asexual reproduction. So the daughter cells are actually clones of the original parent cell there. And like we saw with our prokaryotes undergoing binary fission, the rate of mitosis in this example is rate limited primarily by environmental conditions. So as long as nutrients are plentiful and wastes aren't in excess, then single-celled eukaryotes like yeast will be able to divide and divide and divide through mitosis. But we also see mitosis used primarily in multicellular eukaryotes for cell division as well. So plants and animals both use mitosis as well. And in that case, the purpose of mitosis is not to create new organisms, because most of that is through sexual reproduction, and a specialized population of reproductive cells is required for that. But instead, mitosis is being used to expand out the cell population. So for example, during early embryogenesis, we start with one fertilized egg and we end up with, in an adult organism, tens of trillions of cells. How do we expand out that cell population from one fertilized egg? Well, that happens through mitotic cell division. It's also used to replace cells that have become damaged or worn out or old or that are lost through injury. That is also a mechanism for mitotic division to occur. And once again, just like we saw with our yeast, the daughter cells are going to be clones of the parent cell. So clonal expansion of the cell population during growth and development and replacing worn out cells are both types of functions that are associated with mitotic division in eukaryotic organisms. A third form of cell division called meiosis is used by sexually reproducing eukaryotes to generate their reproductive cells, their gametes. So if we were talking about animals, we'd be talking about sperm and egg here. Those are produced in specialized reproductive organs, the ovary and the testis, from a specialized population of cells called germ cells. So germ cells would undergo a meiotic division rather than mitosis to produce gametes. And this can actually happen in both single-celled and multicellular eukaryotes. So as we learned in the cell signaling unit, yeast are capable of mating with one another, and those cells that would be undergoing the mating and the mating factor signaling would be cells that are produced from a meiotic cell division. But so we see meiosis being used for sexual reproduction in both single-celled and multicellular eukaryotes. But once again, this is going to be limited only to the germ cells in the reproductive organs of multicellular organisms like plants and animals. Now, the important thing to recognize about meiosis is that this is a reductive division. And what's being reduced here is the amount of DNA. And so before any cell division event happens, DNA has to be replicated because we're going to be creating daughter cells, and so DNA needs to be copied. So in both binary fission and mitosis, the DNA is copied once and then divided up once, and so we end up back where we started. But in meiosis, the DNA is copied once, but then the cells undergo 2 divisions that result in daughter cells that have half the genetic material of the original parent cell. And so a cell that has half the genetic material of the starting cell is referred to as a haploid cell. So these are cells that have half the DNA content of the original parent cell, and that's the origin of the term reductive division. It's reducing the amount of DNA. And we're going to talk more about meiosis and the mechanism of meiosis in the next topic, but this is just sort of an introduction to meiosis and how it differs from mitosis. Now, the other thing that we're going to see in meiosis is that genetic diversity is introduced in a couple of ways. So we're going to see that in every meiotic division, we get new combinations of parental chromosomes that didn't previously exist, as well as new combinations of gene versions or alleles that are going to be introduced. So file that away. Genetic diversity is associated with meiosis, and again, we'll look at the details of how that's introduced in a little bit more in the meiotic division lecture. So, obviously, since we're reducing the amount of DNA, we're introducing genetic diversity through meiosis, the daughter cells that are produced through meiosis are genetically distinct and different from both one another and from the parent germ cell that we started with. So we're going to talk more about meiosis later on, but for the rest of this discussion, we're going to focus on mitosis. The first thing we want to do is understand the context in which mitosis is occurring. And we have to understand that the vast majority of the life of a eukaryotic cell is not spent dividing. It's just spent carrying out normal cellular functions that are associated with that particular cell type. Right? And so if we think about the life cycle of a cell, what's commonly referred to as the eukaryotic cell cycle, mitotic phase would represent that part of the cell cycle where the cell is actively undergoing division. But the whole rest of the life cycle of the cell, what we would call interphase, the cell is not actively dividing. All right? The cell may be just carrying out normal cellular functions, any specialized functions that are associated with that particular cell, just sort of hanging out being a cell, or it may actually be preparing for the next round of cell division. All right? So depending on what it's doing, interphase is actually divided up into 3 subphases that are called G1 for gap 1, S phase for synthesis, and G2. So let's imagine that we have a cell that's been produced by a cell division event. So a previous cell division event has produced 2 new daughter cells. This cell is going to be entering interphase, and more specifically entering into this first gap phase called G1. What happens in G1? Well, this cell is going to continue to grow. It's going to be metabolically active. It's going to be expressing genes. It's going to be synthesizing proteins. It's going to be doing anything that is associated with this specific cell type. So, a liver cell versus a bone cell versus a neuron in the brain. Those are all going to have specialized functions associated with them. And then only if that cell is signaled to do so is it actually going to leave G1 and enter the next phase of interphase, which is called S phase. So at this point, the cell is actually preparing for the next round of cell division. And step 1 in preparing for the next round of cell division is to replicate or copy cellular DNA so that the daughter cells produced by that next cell division event are both going to get a copy of that genetic blueprint, that instruction manual for life, right? And so from G1, the cell would proceed into S phase, the DNA would be copied, and from there the cell would enter into gap 2 or G2 phase, which is another growth phase. So final preparations for cell division are going to occur here. The cytoplasmic organelles are going to be duplicated. And importantly, any final repair to the DNA and checks on the integrity of the cellular DNA are going to happen here in G2 as well. Once the cell has double-checked that all cellular DNA has been correctly replicated and any mutations or damage that the DNA has incurred has been repaired, will the cell then proceed into the mitotic phase, which itself is divided up into 2 general subphases called mitosis and cytokinesis. Mitosis is the subphase of the mitotic phase in which the nuclear contents are being divided up, And cytokinesis is the part of the mitotic phase where the cytoplasm is actually being divided up and two new daughter cells are being produced. So those are sequential. First the nucleus is divided up and then the cell is split in two. And as we're going to see, mitosis itself has different subphases associated with it. So the cell has to go through a sequential series of steps to make sure that the nuclear contents are appropriately segregated. Now, before we move on and actually start looking at the mitotic phase and seeing what those steps are, I do want to mention that for some cell populations, once they've been produced and they have become specialized, they undergo what's referred to as an exit from the cell cycle. So that's sort of represented by this arrow here. And they enter what's sometimes referred to as G0, but what's really just G1 forever. So these are cells that are what's referred to as terminally differentiated. So they are specialized, highly specialized cells. And once they become specialized or differentiated into their particular cell type, they can't really undergo any more rounds of cell division without losing that specialized function that they've acquired. So a good example of cells that become terminally differentiated and don't divide anymore are neurons. So as we've talked about before, the extensive connectivity between neurons is really important for the communication and control that's associated with the nervous system to happen. So as you can imagine, if you've got a neuron that's got hundreds of connections with other neurons, if it were to undergo cell division, it would actually have to lose all of those connections, divide and produce two new daughter cells, and then try and reestablish all of those connections. And that would simply disrupt the function of the nervous system and lose all of that highly specialized connectivity that allows communication and control to happen, right? So those cells that exit the cell cycle are referred to as being in G0, but again, that's really just G1 forever. So the cell never gets stimulated to prepare for another round of cell division. All right, so before we start talking about all of the specialized events that go on during mitosis, the division of the nuclear contents, and I should say that we're going to spend most of our discussion on this and not on cytokinesis, frankly, because not as much is known about how the division of the cytoplasm and the cytoplasmic organelles is regulated. So we're going to spend most of our time talking about how the division of the nuclear contents takes place. But before we do that, we need to remind ourselves of what DNA looks like and how it actually exists in a eukaryotic cell, because that becomes very germane to this discussion. So first of all, let's just make sure that we understand what's happening with the DNA as it proceeds through the cell cycle. So let's start with this cell right here, right? That's just been produced from a mitotic event. So two daughter cells were produced, but we're just going to follow what happens with this guy. So let's pretend that this cell actually has two DNA molecules in its nucleus. Those are represented by these straight lines. Obviously, most cells have a whole lot more than two DNA molecules, but this is just to illustrate what happens. So in G1, here we've got this cell, and it's got its DNA in the nucleus. It's two DNA molecules, and those contain the cellular genes that are being expressed and copied and made into mRNA, and that mRNA is coming out here into the cytoplasm, being read by ribosomes and translated to build a protein, as we've talked about many times before, right? So that's what's going on here. The cell's just hanging out being a cell. So if that cell receives a signal, it's going to proceed into the next phase. It's going to start preparing for the next round of cell division. And as I said, step one in that process is copying of the DNA, or DNA replication. We're going to look at the details of DNA replication, the mechanisms that control DNA replication in a whole lot more detail later on. But for right now, we just want to understand that each of these DNA molecules in this cell are going to be copied. And what we're going to have then is our DNA held within the nucleus, but instead of one DNA molecule, we're going to have two identical copies of DNA, right? So each of these DNA molecules is going to be copied, and we now have two identical copies. Those two identical copies are referred to as sister chromatids. So chromatids, TID, are identical DNA copies that are produced during S phase and held together until they're allowed to separate from one another in mitotic phase, right? So these are identical copies of DNA. This individual DNA molecule was copied. Now we have two. These sister chromatids, I'm just going to sort of preview something we're going to see in more detail in a minute, are held together by encircling proteins called cohesins, right? So once the DNA is copied here in S phase, these two identical copies of DNA are held together by these little hula hoop shaped proteins and not allowed to separate until we get to the mitotic phase. So all the way through G2 here, those two copies are going to be held together and not allowed to separate from one another. So the cell's actually keeping them together until it's ready for them to be pulled apart. And remember that what's happening here through the end of S phase and into G2 is that repairs to the DNA are being made. The cell is double checking to make sure the DNA is not damaged in any way. So it doesn't want these two copies, these two sister chromatids, to separate from one another until it's been double checked and triple checked, right? And then they'll be allowed to separate here in mitosis. So again, those proteins that hold the sister chromatids together are called cohesins. So cohesin proteins are ring-shaped proteins that hold those sister chromatids together. As we've talked about before in the course, cellular DNA is not found naked, right? We never find naked DNA in a eukaryotic cell. Instead, what we find is that the DNA is compacted and condensed by its interaction with histone proteins into a form called chromatin. So chromatin is the complex of DNA wrapped around proteins, coiled up and condensed and organized within the eukaryotic nucleus. And so that's what's being represented here. In blue is your strand of DNA, and what we're going to be seeing is the successive levels of increased condensation and compaction that can be associated with the structure of chromatin in the cell. So these little purple squiggly guys are meant to represent the histone proteins that the DNA gets wrapped around, and that DNA again is that little blue strand. So this is a more accurate structural representation of what that looks like. Your DNA is the double helix here in gray, and then these colorful alpha helices here are representing the protein structures of the histone proteins making up the core of that nucleosome. And notice that each of those histone proteins actually has a little squiggly N-terminus tail that projects out from the nucleosome, and that's sort of represented here by these little tails that project out. As we're going to see later on, modification of those histone tails is an important means of regulating gene expression, right? But we'll return to that topic a little bit later on in the course. So the first level of chromatin compaction and organization is just wrapping the DNA twice around a core of histone proteins. Then that actually gets coiled up into this cylindrical structure, sometimes called the solenoid structure. So we go from a 2 nanometer in diameter DNA molecule to a 10 nanometer in diameter nucleosome, and then a 30 nanometer in diameter fiber. These 30 nanometer fibers are then looped and coiled up around a central protein scaffold to form an even larger fiber. So this represents a higher level ordering or condensation or compaction of the DNA. And then these 300 nanometer fibers can then get sort of coiled up and compacted even further. But this level of compaction that we see from 30 nanometer fiber up to this very, very tightly coiled and compacted form of chromatin, we really only see in the context of cell division. In a normal cell that's not dividing, that's in interphase, this would be the structure of the DNA. It would be in this 30 nanometer fiber form. And this makes it easy to sort of unwrap and express those genes in that region of the DNA if that particular protein that gene encodes needs to be expressed in the cell. And so what we're going to see is that at that transition from interphase into the mitotic phase, as the cell is entering mitosis, we're going to see a dramatic shift in the structure of the chromatin from this 30 nanometer fiber structure of chromatin to this very, very tightly coiled and compacted form of chromatin that we really only see in the context of cellular division. And so this is what that would look like. So here's a cell that's actually entering mitosis right now. It's getting ready to divide up already duplicated DNA, and we start to see the DNA change in its structure from a state where we really can't make out any individual DNA molecules like we see up here in the upper right cell. So this would be a cell that's in interphase. We don't really see any distinct DNA molecules, whereas here in this cell that's entering mitotic division, we can make out these distinct little fat fingers of DNA, and that again represents this very highly coiled and condensed state. Now, we talked about sister chromatids before that are produced during S phase. So just to get your orientation here, this would actually be two sister chromatids highly compacted and coiled up and condensed, held together very tightly with one another, again by those cohesin proteins, until they're allowed to separate. Now, those cohesins would actually be all along the DNA. So all along here, we would see those circular proteins sort of holding these sister chromatids together. So initially, the cohesins are found along the length of these duplicated molecules, but they're particularly concentrated here at a region of the chromosome that is called the centromere. Now, the centromere is a specialized region of DNA where microtubules are actually going to be attaching and driving the separation of these duplicated DNA molecules from one another. All right. So at this region called the centromere, we see a particularly high concentration of those cohesin proteins encircling the DNA. And so as the cell proceeds through the mitotic division, these other cohesins that are sort of holding the arms of the chromosomes together, those can actually break and come loose. But this region at the centromere, the two sister chromatids are going to be held very tightly to one another until the cell gets to mitotic division, and those DNA molecules are allowed to separate from one another. So again, here's another visualization of the structural change that we see as a cell enters the mitotic phase. So here's a set of chromosomes as they might appear in interphase. These are G1 chromosomes, and then as the cell enters into the mitotic phase, we go from this very extended, stringy, decondensed state of chromatin to this very tightly coiled and compacted and condensed state of chromatin. If we take a look at what those cohesins actually look like and how they're encircling the DNA molecules, just to get your sense of scale here, remember that I said that that very tightly coiled chromatin structure is actually loops of those 30 nanometer fibers arranged around a central core. So this would be two sister chromatids held very tightly next to one another, and these would be their individual loops of chromatin, right? So blue would be one chromatid, purple would be the other one, and once they're produced through DNA replication, some of their loops are actually interwoven with one another, and that's referred to as concatenation. And it's these loops that would be sort of encircled by those cohesin proteins to hold these two highly compacted and condensed DNA molecules together. So it's not that we have, you know, these humongous, gigantic protein complexes that are encircling the whole giant 1400 nanometer wide chromosome itself. It's that we have these smaller proteins that are sort of at this region where the two sister chromatids are sort of interwoven with one another. So we have these little tiny cohesin proteins in here, right? So that gives you a little bit of a sense of scale. All right, so let's look at the details of mitotic division and the phases of mitotic division to get an idea of what happens as a cell proceeds to divide up its DNA. Here we're starting in interphase. So this is a fluorescence micrograph a light microscopy photo of a living cell in which two types of proteins have been labeled with fluorescence. The first protein here in orange is the histone proteins, and so those are the ones that are associated with the DNA molecules. And so wherever the histones are, DNA is wrapped around them. So that's going to allow us to track what happens with our DNA. And then in green, we have fluorescent labeling of the microtubule network in the cytoplasm of the cell. And as we talked about before, microtubules have an essential function to play in the cell in that they form what are essentially superhighways within the cell that motor proteins can transport materials along. And during mitotic division and meiotic division, the microtubule network serves as the transport track for the DNA molecules that are being segregated and sent to two different daughter cells. So what we see as the cell enters into mitosis is a massive reorganization of that microtubule network. So the green fluorescent labeling is going to allow us to visualize that reorganization as well. So here we're starting with a cell in interphase. It's not actively dividing. You can see that its microtubule network is sort of in this loosely interconnected state throughout the cytoplasm. But then as this cell gets that signal to start preparing for division, it's going to copy that cellular DNA. And once it's gone through G2 and verified the integrity of the DNA and verified that the cell has grown enough and that the cytoplasmic organelles have all been duplicated, it's going to proceed into mitosis. And the first subphase of mitosis is referred to as prophase. So prophase is the first subphase of mitosis. And what happens in prophase is that the DNA starts to condense and go into that very, very tightly coiled and compacted state that we were just looking at. All right, and then the cell enters the next subphase of mitosis, what's called prometaphase. And in prometaphase, a couple of things happen. We start to see the microtubule network being reorganized, extensively reorganized from this sort of network-like structure into a structure that's referred to as the spindle apparatus that you can get a better look at in the next phase here. So massive reorganization. And that reorganization is carried out and organized and regulated by a structure that you can sort of see here, this sort of circular structure, and that is the centrosome. So the centrosome in animal cells is the microtubule organizing center. It's where the microtubules sort of originate from and where their shape and orientation is directed from. All right, so there are different types of microtubule organizing centers and different types of cells. In animal cells, it just so happens to be a centrosome. And what we would see inside the centrosome is two bundles of microtubules called centrioles, and that's where each of these microtubule fibers is actually originating from and arraying out from that centrosome in this very star-shaped pattern. Right? So the cell has two centrosomes that are here very close to the nuclear envelope. And in prometaphase, what we start to see is that these centrosomes start moving apart from one another so that they end up on opposite sides of this nucleus, and they reorganize the microtubules so that they're sending out this radial array of microtubules from the center of the centrosome. And as the nuclear envelope starts to fragment, so that double phospholipid bilayer structure starts to be disintegrated, these microtubules start reaching in and forming attachments to the DNA molecules that were present in the nucleus but that are now exposed. So let's zoom in and take a closer look at what happens during prometaphase when those microtubules start reaching out and making their attachments to those sister chromatids in preparation for separating them and sending them to two daughter cells. So this diagram is meant to represent one chromosome that is comprised of two sister chromatids. So these squiggly bits here would be the sister chromatids. And these ring-shaped proteins here in the middle would be our cohesin proteins. that we talked about before that are holding our sister chromatids together and preventing them from inappropriately separating too early. And you'll recall that we said that the highest concentration of those cohesin proteins is going to be in a region of the DNA referred to as the centromere. Now, the centromere isn't only characterized by the fact that it has a large concentration of cohesin proteins there, it's also the site where we see assembly of a protein complex called the kinetochore. So here in red, I'm just drawing a blobby set of proteins binding and assembling on the DNA in this region of the DNA called the centromere. And this set of proteins, this protein complex, is called the kinetochore. Right? And if you break this down, this kineto is referring to the fact that this is where the separation or movement of these chromosomes is going to be facilitated. Because what's going to happen is that we're going to have our centrosomes out here in the cytoplasm. So centrosome here, centrosome here. And these are our microtubule organizing centers, and each centrosome is going to send out microtubules. And there'll be about a dozen or so microtubules that reach out and connect up with this complex of proteins called the kinetochore on the centromere of each sister chromatid. And I'm only drawing a few lines here, but there would be about a dozen or so microtubules connecting up to each kinetochore. And if you recall from earlier in the semester, you'll remember that the way that transport happens down these microtubules is through the activity of motor proteins. So we have to sort of imagine these little motor proteins that are walking along these microtubules, and they're going to be connecting up with these kinetochore proteins. And so when the cell is ready for these two sister chromatids to be separated from one another, these motor proteins are going to be pulling in opposite directions towards the opposite poles of the cell from which the microtubules are radiating. But this complex of proteins that gets assembled on the centromere of the DNA is called the kinetochore, and that's what these motor proteins are actually making their attachments to. Okay, so this happens in prometaphase. These microtubules start reaching out and making contact. These motor proteins start connecting up with the kinetochore proteins, and they're trying to pull, right? They're trying to pull these chromosomes apart from one another, these chromatids apart from one another, but they can't separate them. Why? Because these cohesins are still intact. And are preventing these chromatids from being separated from one another, right? And so essentially what you get is this tug of war where these motor proteins are pulling that way, these motor proteins are pulling that way, and nobody's really making any progress. And that generates force on these chromosomes, right? And so when everybody's attached up to the kinetochore and motor proteins are assembled on either side and pulling equally in opposite directions, then we end up at a stage called metaphase, where the chromosomes here in orange are actually aligned at the midline of the cell. And what we can't see here are the motor proteins that are trying to tug them apart, but they're sort of at a standoff because the cohesins are preventing the separation of these DNA molecules. Once the cell has detected that equal tension is present on each of the sister chromatids, that is, that they are correctly connected up at their kinetochores to the microtubule network, then the cohesins will be cleaved. And as soon as the cohesins are cleaved, there's nothing preventing these motor proteins from being able to pull. And so that's exactly what's going to happen. These motor proteins are going to start pulling in opposite directions, and we're going to see the sister chromatids, again here in orange, being pulled apart. And so when we see that the DNA is being segregated and pulled to opposite poles of the cell, we say that that cell is in anaphase. Following anaphase, what we're going to see is that the nuclear envelope is going to start to reform around this set of DNA at each end of the cell. And then ultimately what we're going to see is cytokinesis, where the cell membrane gets pinched in an ever tighter circle between these two ends of the cell, effectively pinching off the cell to form two new daughter cells. And this is what that cytokinesis, or separation of the cytoplasm, looks like. So here's an electron micrograph showing a cell undergoing cytokinesis, and what we can see is that the plasma membrane is actually being pulled in and forming this furrow, referred to as the cleavage furrow. And so what's happening there is something that's very similar to what we see happening in muscle cell contraction. We have microfilaments, in this case a microfilament ring, that gets pulled on by motor proteins in such a way that the ring of microfilaments gets smaller and smaller and smaller, and the ring of microfilaments is actually attached to the plasma membrane, and as that ring gets smaller and smaller and smaller, it pulls the plasma membrane in, and we see effectively a pinching off of the plasma membrane, resulting in the separation of the cytoplasm and two new daughter cells. Now, as I said at the beginning, obviously there has to be a very, very tightly controlled set of steps that are ensuring that the Golgi apparatus is duplicated and segregated appropriately to these two daughter cells, that the smooth ER and the rough ER are segregated appropriately. We're not going into any of those details because very little is known about how that's accomplished. We know a lot about how the DNA is divided up because that's something that you can observe very easily just using a light microscope. So people have been, looking at that for a really long time, but the technological tools to investigate how Golgi separation and separation of those other cellular organelles is happening have only recently started to become available, and so that's a very active area of research. So what I want to do now is show you actually a video of some cells undergoing mitosis so that we can see what this looks like in real time, because it's not actually a set of discrete steps. It's one continuous and very dynamic process that once it begins sort of flows from beginning to end in a very dynamic fashion. So what we see here are two kidney epithelial cells, and this cell up at the top is going to stay in interphase. It's not going to be dividing, but this cell down here at the bottom is getting ready to undergo mitosis. And so we're going to go through this a couple of times, but I want you to pay attention to what's happening here in the nucleus. Again, we're seeing that the histone proteins that are complexed with the DNA in chromatin are labeled with fluorescent orange, and the microtubules are labeled with fluorescent green. And so you're going to see, as this cell enters mitosis, the highly compacted form of DNA starting to form, so those chromosomes that are highly compacted becoming visible. And you're also going to see that this cellular microtubule network gets massively reorganized by these centrosomes that are present right here at the nuclear envelope. So let's watch this a couple times, and I want you to pay attention to what happens with the DNA because we're going to see something very interesting. Okay, so let's watch that again. I'm going to pause it at key points here. So we've seen the chromosomes condense here. You've seen the microtubules get reorganized. And what we're seeing at this stage is that the microtubules are starting to form their attachments to the kinetochores and starting to tug on these chromosomes. And that's why they're wiggling and wiggling and wiggling. And you can see pretty early on that the chromosomes get connected up correctly, so they're being tugged from opposite directions. But look at this guy down here. You see what's happening with this DNA molecule? It's not connected up correctly. It's being pulled only from this centrosome, but not from this centrosome. And so the cell actually has to wait for that DNA molecule to get connected up correctly before it proceeds to anaphase and telophase and the rest of cell division. So we'll watch it one more time. So tugging, 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 microtubules connecting up to kinetochores, and motor proteins pulling, pulling, pulling. And everybody's getting connected up and pulled to the midline of the cell equator, but the cell doesn't divide yet because that chromosome was not connected. As soon as it gets connected up and tugged to the middle, the cell gets the signal that equal tension is present, all chromosomes are correctly connected, and the cell proceeds to anaphase. Okay, so as you can see, the process of mitosis is very dynamic, and once it begins, it sort of flows from beginning to end in a very smooth and dynamic fashion. As soon as, you know, certain molecular events have been accomplished, then the cell is going to move on to the next stage, and to the next, and to the next, and ultimately to cytokinesis, resulting in the separation of those newly produced cells into distinct cells from one another. So even though it's really useful to divide up mitosis into these subphases, prophase, prometaphase, metaphase, anaphase, telophase, etc., in some sense they're kind of arbitrary distinctions and boundaries, right? They're ways to describe the events that are happening during mitosis in a more coherent fashion, but it really flows from beginning to end in a very, very nice, smooth fashion. I've given you a link in the PowerPoint slides to another video that's kind of fun to watch. There's a bunch of grad students sort of doing an interpretive dance of what mitosis might look like using pool noodles. So check that out. It's kind of funny. I won't show it to you here. So let's move on and actually start thinking about mitosis as it would happen in an animal cell. So we've been looking as it would happen in a plant cell. We've been looking at animal cell mitosis. So what would happen in a plant cell? Well, it turns out that the basic mechanism of mitosis is exactly the same as what we would see in an animal cell, and the subphases of mitosis would be identical. So we would start with a cell in interphase. Oops, let's change that ink color. We'd start with a cell in interphase. Right, so this is a plant cell just hanging out being a plant cell. If it gets a signal that it needs to divide, produce more cells in this localized region, we're going to see duplication of the DNA exactly the same way that we talked about. We're going to see, you know, checking that DNA replication is complete, DNA repair has been accomplished, going through G2 and then into the mitotic phase. And the first subphase of the mitotic phase is going to be prophase, prometaphase. I'm just going to call that prophase here, where we would see condensation of the chromosomes, fragmentation of the nuclear envelope, reorganization of the microtubules into a spindle-like structure. Now here's one of the differences that we would see in plant cells. You can't really see it in the microscopy image here, the difference in the microtubules. But plant cells, it turns out, don't have centrosomes, and so they organize their microtubule network in a different fashion. It actually relies on a ring of microfilaments that are here at the midline of the cell, and so organization of the microtubule network is a little bit different. But it does get reorganized, and motor proteins are still going to attach up to the chromosomes. We're still going to see that tug of war, tug of war, tug of war, and eventually we're going to see alignment of the chromosomes on the cell equator, what's sometimes referred to as that metaphase plate. It's just the midline of the cell. And then once the cell has received the signal that equal tension has been accomplished, everybody's lined up in the middle, those cohesin proteins are going to get cleaved, and we're going to see anaphase, resulting in the separation of our sister chromatids being pulled to opposite poles of the cell, and then telophase, in which new nuclear envelopes are reformed around these two sets of segregated DNA, and eventually we see cytokinesis, which divides this one cell into two daughter cells, each with its own nucleus and its own set of genetic instructions. Now here's a second difference that we see coming into play here at telophase and cytokinesis, sort of combined here at the end. And that's that, if you'll recall, we talked about this before, that plant cells have a cell wall comprised largely of cellulose. So in cytokinesis, it's not enough to just pinch off the plasma membrane. These two cells, if they are to be distinct cells from one another, actually have to have new cell wall built between them. And the way that that's accomplished is that the plant cell will lay down this little row of vesicles, and inside the vesicles what we see is cellulose and other components of the plant cell wall. And so we see this whole layer of vesicles laid down the middle, and then as those vesicles fuse with one another, what we end up with is plasma membrane extending from one side of the cell to the other, and then in the middle what we're sort of illustrating in orange here. This would be what was inside the vesicles, which is the cellulose and other cell wall components. So that's referred to as the cell plate, and you can actually see that very nicely in microscopy images using a stain that will identify cell wall components, cellulose and other molecules that would be in cell wall. So those are really the only two differences that we would see in plant cells versus animal cells. If you're interested, I've given you a link in the PowerPoint slides to a very nice review article that talks about how the plant spindle is formed, and it gives you some details of the molecular mechanisms that allow plant spindle formation in the absence of centrosomes, which is something that's only really been understood in the last 5 years or so. So this is a relatively new area of research. So in this video, we've gotten an introduction to 3 different mechanisms of cell division that are used in different contexts and different organisms. We've seen how binary fission works. We've taken a look at the molecular events that occur during mitosis. We've compared mitosis in plant and animal cells. In the next topic, what we want to talk about is regulation. So in the context of a eukaryotic cell in a multicellular organism, like an animal, how is cell division regulated in a very tight and coherent fashion so that cells only divide when they're told to and behave like good citizens in the context of the organism as a whole?